Basically, when we talk about a sarcomere, that is the length of one contractile unit from Z disk to the next Z disk. It is at this sarcomere that everything really goes. All right. The M line contains the protein desmin. And as you can see where the M line is, it's sort of the end point. Okay. The M line is the anchoring site for all of the myosin filaments. The Z disk, okay, is basically the anchoring site for all the thin filaments, all the actin filaments, okay? So you have the A band made up of the myosin filament. You have the Z disk. It is made up of protein connectin. It's in the middle of the I band, and it's the end of the sarcomere, okay? The I band is the free zone of contraction for myosin and actin. They're going to basically contract and roll over each other, but I'll show you how that works in a while. The elastic filaments, which you will see in a little bit, these make the protein tighten, which allows myosin recoil during the relaxation. Let's see if I've got that next one. There we are. If you look at the yellow, that's the elastin. That allows for a recoil, a relaxation, so that they can all get pulled apart after they have been attaching. Notice that the big red area is called the myosin, the thick filaments. They are going to attach to the thin filaments. These are the blue lines, and they're made of individual little proteins called actin. They're like beads on a string. They're attached to each other, okay? The thick filaments are surrounded by a hexagonal array of thin filaments. If you take a look at the I band, that's thin filaments only. Take a look at the H zone, that's thick filaments. You have the M line, the thick filaments are linked by accessory proteins. Now look at the outer edge of the A band. Notice that the red dots, the big red dots, are the myosin filament. Around them are all these actin filaments. So the heads of the myosin will attach to any one of the surrounding actin filaments that is available and that is close. And they will begin to pull and move forward. Okay, and we'll get more of that in a bit. When we talk about the thick filaments, they're composed of myosin molecules. Myosin, if you take a look at the lower left-hand side, is really this really unique little protein. It's got a rod-like structure, and they come out to a tail, and it has two globular heads at the end, okay? And it has a flexible hinge region. That's going to be key to moving, attaching, ratcheting forward, etc. It has on the heads an actin binding site and an ATP binding site. ATP has to play a key role in providing the energy for the ratcheting forward. Actin will basically be the site where the myosin heads attach to it. Okay? Now, when you go to the thin filaments made up of actin subunits, and you see these subunits down in the lower right-hand corner, you see they're all kind of like little beans. On the actin subunit is a binding site for myosin attachment. And then what they have are these two kind of strands that wrap around each other for the actin thin filaments. Now, don't get that confused about the wrapping around with the tropomyosin and the troponin. I'll give you some info about that in a second. But what you have to understand is this, that initially, if you look up at the thin filament here, it's just basically two strands, each composed of little beads of actin, but the strands wrap around each other and they expose in different angles the myosin binding site. And then you see the myosin heads that will attach Now, this is a two-dimensional view. We're not talking about the hexagonal array because it would be kind of a lot more difficult to see it from a three-dimensional aspect. But the videos should provide you with some of that three-dimensional aspect. The other key point that you want to understand is this. There are two very important proteins that also play a role in this process. One of the proteins is troponin. We'll get into that in a second. The other one is a big, long, wiry type of protein called tropomyosin. Tropomyosin is sort of the boulder on the train tracks. It blocks by actually physically coming and standing or lying right in front of the active site of the actin subunit. So it blocks myosin from binding, but it has to be moved aside so that the active sites for myosin attachment on the actin strands will actually be able to have the binding. In other words, think about it this way. Like I said, the boulder that's on the train track, well, the train's not going to hit the boulder. It's just going to stop. So if you move the boulder over, in this case, you move the tropomyosin over, you will then have freely exposed actin sites for the myosin to attach to. But what is able to move this? This is where we get into the other protein. Troponin is a protein that contains 3 units. So it's got a triplet. It's got a binding site for tropin, myosin site, a binding site on the actin protein. It's got a binding site for calcium ions, which play the key trigger. And then it has a binding site for tropomyosin strands. So you've got TNI, the first protein. It binds to the myosin binding site of the actin protein. You've got TNC. These are all troponin, but they're basically made up of a triplet. And you got TNC, and it binds to calcium ions. And then it's got TNT, which binds to the tropomyosin. So to give you it in a short flashbulb version, calcium comes in, it binds to the TNC. As a result, that causes the TNT that blocks the actin binding sites. It causes the tropomyosin to be pulled away so that actin can now be bound by the myosin. Okay? Now there is a protein that anchors all of these filaments to the sarcolemma. And that protein is called dystrophin. And we'll get into this later on, but this is the protein that has a mutation that leads to a particular type of muscular dystrophy. And we'll explain that more in a bit. Here you see an electron micrograph of the myosin heads, and they are attaching to the thin filaments. So myosin, which is the thick filament, its heads will bind to the thin filament, actin. Now, I refer you as another possibility to hot page 286, I think it is. It's table 9.1 in your text. And basically, if you use that as a quick review, it's going to help you because it's going to go from a structural organizational level of macro or gross anatomy right down to the micro. And that would be very helpful for a quick review. Excuse me. Let's deal with the sarcoplasmic reticulum, which is a modified smooth endoplasmic reticulum that we talked about, and these unique structures called T-tubules. Okay. Now, as I said to you before, calcium is the key to muscle contraction. Now, there's a nice bit of documentation in your chapter, and it's a focus and says the excitation, contraction, coupling. Read it, review it. It'll help you to understand. What you see here is a nice magnification of the skeletal muscle fiber cell. And you can see there sarcolemma, the ample number of mitochondria that are going to supply ATP. you're going to see the myofibrils, and you can see remember the red dots and the blue dots. Remember, red dots are the thick filaments, blue dots are the thin filaments. But then you're going to see what's called the triad, which is T tubules and two of the terminal cisternae of the sarcoplasmic reticulum. This is all part of the signaling to cause the muscle contraction. And we'll explain that, okay? Keep in mind that sarcoplasmic reticulum is an elaborate network of tubules that surround each myofibril. You see how they kind of spread out and they kind of branch and come together and branch and spread out. That's because what they're going to literally do is dump, and basically you would call it coat or salt or whatever you want to use for your analogy. They're going to basically saturate those myofibrils with lots and lots of calcium so that all of them will begin to contract. Now, by the way, it doesn't always have to be an all or nothing, and I'll explain all that as we get into it. But just to get the gist of it, just to get the beginning, you get this big discharge from the terminal cisternae. They have cross channels occurring in pairs at the AI junction. Notice where the AI junction is? That's where the A band and the I band come together. And they lie right next to the T tubules. The function of the terminal cisternae is storage, reuptake, and release of calcium ions to trigger contraction. That's a pretty straightforward statement. In other words, yes, they will release calcium. Yes, they will take it back up. And as they do that, that stops the contractile process. The T tubule is very interesting. It's a sarcolemma tubule that passes into and through the cell. Think about this, comes right up to the level and comes right up to the edge, and it's almost like a tube right attached to the surface of the sarcolemma, and it goes all the way down. Now, the T tubule, excuse me, is located next to the AI junction. This T tubule will carry the membrane depolarization from the cell surface, and which is the muscle contractile signal, to the sarcoplasmic reticulum to initiate the sarcoplasmic reticulum release of calcium. Now, you can see this a little bit as we go into the contractile process from fully relaxed to fully contracted. Notice the striations and how they change. Notice the lightly colored bands and how they disappear. When you have a fully contracted sarcomere, the thick bands basically take up most of the space throughout the sarcomere. Okay? You can see this up close. Notice the heavy staining here. This corresponds to basically actin, myosin, actin, myosin, actin, myosin. That's fully relaxed though. When it's fully contracted, you can see that the myosin has traveled much further deeper into the area where there is actin filaments. And so basically a very little lightly stained area, which was composed only of actin. You've got lots and lots of actin myosin overlap here, okay? Now we've got to get into the contraction, which is a process. This is a sort of a chart, and it's kind of hard sometimes because what you want to do is give the sliding filament theory of contraction, but then you've got to go back over the phase one, which is the muscle fiber is stimulated by the motor neuron. Hang in there, we're going to go through some of this. One thing I want you to be mindful of, the nerves, as we said earlier, will be the trigger to cause the muscles to contract. The nerves will have these terminal buttons, or they're called end plates, that they release a neurotransmitter onto the surface, the sarcolemma, of the muscle. When they do this, this starts up a short little depolarization. And this is where we have to go back and review. Voltage gated, ligands, and